discovered in 2000 after a 63-year period with no confirmed sightings. Since rediscovery, the species has been observed breeding at 5 locations, 3 along the Chinese coast in Zhejiang province, that's south of Shanghai, facing the East China Sea, and 2 other small breeding colonies were located off South Korea and Taiwan. The total population size was estimated at no more than 50 individuals, and the species is, of course, critically endangered. In 2013, a tern restoration project was initiated in Jiushan Islands, and the location of a former breeding colony of Chinese crested tern that was abandoned in 2007 after breeding failure due to illegal egg harvest, which seems to be a big problem threatening these terns. And later on, the restoration project was extended to 2 other islands of the Suzhou group. Since their rediscovery, Chinese crested terns had only been found nesting within large colonies of another species, the greater crested tern. You can see pictures online and in the paper of Chinese crested terns among these greater crested terns. So the idea of the authors was to use social attraction techniques to try and establish new breeding colonies of greater crested terns, hoping that Chinese crested terns would be attracted by those colonies and would recruit to them. So what they did at the restoration sites is that they cleared the islands of rats and later of snakes. They also cleared vegetation in a rectangular area about 60 by 200 meters to make space for a prospective tern colony. And they also guarded the islands across the breeding season, which gave them the opportunity to have observers to check on the birds. And all of these people well, the teams on the islands deployed decoys across a prospective colony site, and they had playback systems playing tern calls. So these are standard techniques which have been used successfully to restore breeding colonies of nearly 50 different seabird species around the world. And after a lot of effort, this all worked. So that means that in 2013, it took about a month and a half for a group of terns, both greater crested terns and Chinese crested terns, to respond to the decoys and the playbacks and land onto the new site. And in subsequent years and breeding seasons, both species started attending the sites right from the breeding sites right from the beginning. In 2015, the minimum total number of breeding adult Chinese crested terns reached 75 individuals, and the authors now think that the global population of Chinese crested terns exceeds 100, which is still small, but already much better. So overall, this project was the first attempt in China at active restoration of a seabird population, and it made the news, notably through a 4-episode documentary film broadcasted by China Central Television, and that really helped raising the profile of the project and preventing illegal egg harvest. The authors think that their approach should be expanded spatially to build a conservation network, including migratory stopover sites and wintering sites across the Australasian flyway, and as well as at breeding sites in the East China Sea and the Yellow Sea. Yet, these terns, they apparently face a new challenge because molecular studies have now shown that there's hybridization between the Chinese crested terns and greater crested terns, and the hybrids seems to be fertile. So new challenges ahead. So congratulations on this effort and the success at restoring these tern breeding sites. I really enjoyed And I felt the excitement also of the authors while reading the paper. That was really noticeable. And I happen to have witnessed a similar constellation in India together with my PhD student Ravichandra Mondretti, who I think is online. In Lakshadweep, there's also a problem with illegal egg harvesting at Nodi and tern colonies. And I noticed that the socio-economic context was also quite important at explaining why and how people take eggs. So for this situation in China, I was curious about the context there. So the people who try to take eggs illegally, do they really need these eggs as a food supply, or is it more a delicacy which comes on the top of already available food supplies? That would be my first question.
Okay. Trying to, I think we've lost Yue. He seems to have dropped off the call. So I'm just trying to track down a couple of the other authors of the paper here as well. We've got, there we go. Xi Wang is on there, and Shui Hua Shen is, and Zhang Yong as well. There we go. Okay. I guess, David, who would you like to direct that question to?
Yes, so I'm happy to repeat the question. My question is about the socio-economic context of the study, and specifically whether people who try to take the eggs illegally, do they really rely on that on these eggs as a food source, or is it more a delicacy?
Okay, maybe I can answer the question. I think the poacher, the egg collector, not rely on this as a food. I think most of the collector, they didn't realize this is illegal. They didn't realize the harm to the seabird. So I think the education is more important to the close, along the close line.
Yeah, that was also exactly my impression in India, that people just didn't know that they shouldn't be taking these eggs, and they didn't realize the impact it would have on the birds. So in that sense, the restoration project you have been running so successfully, is it linked also to education programs at the regional scale to make people more aware of the impact of egg harvesting?
David, are you still there? Oh, you are. Yep.
Yes, I thought there was a long lag in my question. But
perhaps if you direct the question to somebody, David, might help.
Yes. So Xuan, I was wondering whether you had an education program linked to the restoration program to make people more aware of the impact of egging.
Yeah, we have the special education program, and for the especially in the area and nearby the breeding island. We also have some education program along the all Zhejiang coast. I think that in the future, Asa Island may be the potential breeding habitat for this Cretaceous species.
And since this is such a rare species, did you get invaded by birders who wanted to twitch that rare species?
Pardon, maybe Dan can some explanation.
Because that can be a threat in itself.
Yeah.
Dan?
Yeah, there was some of that, as Shaewon knows, but not near as much as on the Matsu Islands.
Yeah. Yeah, I can.
And I'm curious about the potential competition between these terns and local fisheries. Do you think that this could also be an issue limiting the populations? Hello?
I think we had some interference there. There's something got unmuted, and there was a lot of wind on somebody's microphone.
Yeah, the question was whether you think that the tern populations could be also limited by local or regional competition between seabirds and fisheries.
I don't think the seabird competition is the limit for the Chinese crested tern. I don't know if my answer is.
No, and I was also wondering. So this you doubled the estimated population size. So the estimated population size for the species went from 50 to 100. Do you think that it's through improved observation of a population which was already existing, or do you think that already in about the 10 years of the study, there have been many more recruits into the population?
I'm not very clear about your question. Maybe Dan, could you? I don't know if that.
Yeah, I think it's probably a combination of both, David. I think there was clearly not a it wasn't well understood where all the Chinese crested terns were when this project started in 2010. But I think some of the birds that were nesting places that Shaewon did not know about have gravitated to Taitandao and the Wuzhishan Islands because nesting success has been relatively good there. But I also think that there's been such good productivity at Taitandao and Wuzhishan Islands in the last 6 years or so that that's been a big factor in the increase.
But the overall population is probably still so sensitive that you're not banding or doing anything in that sense?
No, as a matter of fact, they just got done banding well over 200 chicks on Taitandao. Very successful, including five Chinese crested terns.
Okay, so in a few years, you may know whether there are new recruits.
Yeah, well, we know from banding that has been done at the Matsu Islands that at least one Chinese crested tern fledgling came back to the breeding colony on the Matsu Islands the first year, which is extraordinary. I mean, for many other terns, the young of the year don't return to the colony at all, and they don't return until they're 2 or 3 years old.
Okay, no, very interesting, and congratulations on this study and all the efforts. which it has costed. So I still have one or two questions, but I wanted to hand over to Grant and also, of course, to the audience in case there are questions.
No worries. Thanks, David. Yeah, and
Thank you.
congratulations again for this paper. It's this obviously this work is very important, particularly for this species. The question that I have actually, Johannes Fischer just wrote down in the chat box, which is basically if hybridization is a threat, would it be more beneficial to directly attract Chinese crested terns rather than the greater crested terns? Shua, do you have an idea of that? Is it possible to just attract Chinese crested terns, or is it a problem because the population is so low that you have?
Yeah.
So
yeah, I think it's for the first time we found that the Chinese crested tern. This species is breeding within the great crested tern, and we think it's very difficult to just attract the Chinese crested tern. So if I think it's more chance we attract the crested tern to breeding on we selected island, and the Chinese crested tern can follow the great crested tern.
mm-hmm. Yeah. Do you think that with given time, as the population potentially increases, that you could get to a point where you could focus only on Chinese crested terns?
Yeah, I think. Yeah, sure, yeah.
The other question I had too, which actually goes back to what David asked the first time about the threats from egg harvesting. How many people are engaging in egg harvesting? Is it a lot of people, or is it a very select few?
I don't think there are a lot of people engage in the egg collection. I think just some fishermen, yeah, a little part of fishermen. They occasionally they find the breeding colony. Yeah, they will, and most of the time they just fish collecting. Yeah, and then they will because there are so few seabird breeding islands along the coast. So once they find the breeding colony, they will land and collecting the egg.
Right. Thank you. We have a question from Eric Voller. I'm going to unmute you, Eric. Oh, actually, you can probably unmute yourself. I've got that set. Eric? Where'd you go, Eric? Oh, there you go. Okay, Eric, I'm unmuting you. Maybe.
Unmute it.
There you go. Gotcha.
Can you hear me?
Yeah, got you fine, Eric.
Good. Shaywa, first, I want to thank you for your efforts and your collaborators for your remarkable efforts to conserve a threatened species. It is a remarkable effort. I am interested in the issue of hybridization between the two species. Do you have an idea why there is now a prevalence of hybridization when the two species have occurred together in the past and not hybridized?
Why? I think maybe there are two reasons for the hybridization. One is they are too close. The two species too close relationship, and the other is I think it's the hybridization is occurred in the late breeding phase. Some individual they can't find the mate, and they will find the SS species, yeah, as their mate. So it's the so there's very low percent. I think maybe no than 5% this occurred.
Okay, thank you. Yes, because I work on fairy terns and little terns here in Tasmania, and we see occasional hybridization between the two species, but we have no evidence of the hybrids being fertile or viable. So it's an interesting situation where you're seeing hybridization and they appear to be fertile. So that was the basis for my interest in your work.
Besides the Chinese crested and the great crested tern, we also found that little crested tern also have hybrid with the Chinese and the great crested tern in the same island.
Okay. Okay.
Thanks, Eric. I've got a question from Ravi. Ravi, I'm going to unmute you. Hang on a second. Okay, you're unmuted. Go for it. Oh, Ravi, I can't hear you.
Sorry, now you can able to hear me?
Yeah, can
Hello.
hear you now.
Yeah. So I just have a congratulations on your study. I have a question. I know I want to add upon David regarding this, especially a question on socio-cultural context. So when we're working in Lakshadweep archipelago, so we found a link between tradition of, for example, between the social tradition of egg poaching. So people have been collecting eggs over, for example, over decades. So this has been a tradition. Did you find any similar practice in your study area? So that was one question. So hope I'm audible and clear. Yeah.
I'm sorry. I'm sorry for my English. Maybe Dan can.
Do you want me to? Do you want me to repeat the question, for example, so I can? Yeah.
Yes, please.
Yeah. So this is a socio-cultural. My question is, pertains to the socio-cultural context. So it is of the practice of egg collection. So, for example, in my study area in Lakshadweep archipelago back in India, so what I found is they have been collecting eggs as a tradition. It's part of their custom, social custom. So it's been going on for ages, and they know that the terns get affected by this collection. So people do know that. Did you find any similar traditional practices? For example, any link to the tradition in your study area? So that was my question.
Was that a little bit clearer, Shiwai?
Hope I'm clear. Otherwise, I can type. I can type it out for you.
No. I think we can translate that relatively easily. Basically,
Yeah.
the question, if I'm understanding it right, was in India there are traditional practices associated around egg collection. So it's a traditional practice rather than some fishermen going out and collecting eggs for food. So what he's asking is egg collection of the Chinese crested tern eggs, is that a traditional practice, or is that simply a social construct from recent times?
Yeah,
I think it's a traditional. Yeah. Along it's
Okay. Does that
okay.
that answer your question all right, Ravi?
Can I say something?
Oh yes, please.
Yeah, sure. Yeah, please. Sure.
Oh, can you hear me?
Yes.
Okay. I just want to add something to this answer, because when we were doing breeding seabird survey along the coast on the island, the fishermen they knew they know that after egg collecting, the terns will leave. So they know this egg collecting will affect the tern. But I think most of the people, they will just let it go. Well, when people see there are some people collecting eggs, most of the fishermen nearby, they will just go past them, and they won't stop them, because I think many people still think that it was normal to collect eggs, because the local people, they will say that their grandfather or great grandparents, they have been doing this all the time. But they will also tell us that the terns are fewer and fewer, and until there are almost no eggs on the island, and you can hear them say that these islands used to have a lot of terns, but they just
One thing we tried in India in this context, we tried to explain to the local fishermen that they need the terns as indicators on where the fish resources are at sea. And so that was a way we were trying to convince them to stop collecting the eggs, because if the terns are gone, they'll lose their best fishing indicators when operating at sea. I wonder whether this might be also a useful argument along the Chinese coast.
Can I can I answer the question? Okay, I think as like Dr. Chen just said that just a few people will collect eggs along the coast. Most of the people, I think most of the people, they even don't know that there are people collecting eggs. Because most of the people who live in the cities, they haven't they don't know there were people land on the island to collect eggs, but in the local area, some old people, they will do this. Just a few people, but because the breeding colonies are not very there are just a few breeding colonies along the coastline, so so the effect is sometimes it's big. But I think most of the breeding colonies will try the second time or the third time for breeding, and I'm not sure if the fishermen see the terns as like a competition or something. They don't care the terns because the terns only eat small fishes. It won't. It doesn't have much effects on their harvesting.
Yeah, I see what you mean. Where it has no direct effect, but if the terns indicate small fish, this can be in turn an indicator of larger predatory fish going after the small fishes. So that's why the terns could be good indicators. But yeah, I don't know whether it works in that specific region where you are working.
Great. I think we should probably move on to the next paper now. It's 10 after 8
Yes.
my time, so we're getting a little bit over time, but that's okay. Thank you very much for that that paper. Thank you for
Congratulations on the study. Yeah?
Thank you.
Yeah,
Thank you.
congratulations!
Thank you.
Congratulations on all the hard work. It's obviously
Thank
very important, and we're very, very honored to have you here today. So I think with that, we'll move on to our penguin paper. And thank you. I hope you do stay and listen to the discussion over this
Sure,
weekend.
sure.
Thank you very much.
Thank you. Thank you.
Okay, we've got another paper coming up. We've got Michelle Villa on the line. Hello, Michelle. I'm going to unmute you. Let's see if this works. Hey, there we go.
Hello.
Hi, Michelle. How are you?
I'm good. How are you?
I'm good. It's been a long time.
For a very long time.
How have you been?
Oh, good. Can't complain. I'm in Melbourne, so we're in lockdown 2.0.
Listen to you. You've even got an Australian accent now.
True. Well, it has been almost 4 years.
All right, let's get on with this. I hope you all get a chance to look at that YouTube link I posted at some point, because you know, obviously, because penguins are fake, none of this matters. So yeah, another paper on penguins. I guess this is our third week in a row with Antarctic penguins, which is it's not a bias, I promise. It's just it just happens to be that way. So it's estimated that Antarctica broke away from South America over 180 million years ago. So it's kind of unsurprising that animals who evolved in Antarctica are generally quite isolated. Not only are they isolated geographically, but they're also exposed to some of the most extreme environmental conditions on the planet. And it's because of this isolation and exposure to these extremes that it's kind of previously been thought that penguin species are simply not exposed to many viruses. I guess it's kind of been thought that it's been so miserable there that even viruses don't want to be there. The reality is that the methodological techniques to quantify viral diversity were not really available until somewhat recently. Kind of up until lately, most studies quantifying the viruses in birds had to rely on viral species whose genomes had already been sequenced. And the ones that were most readily available were kind of from chickens, basically. So we were kind of comparing everything to chickens, which isn't bad. I mean, I've got 22 in my yard right now, which are right outside my door. So I could probably send you some samples, Michelle. This particular study, which was led by Michelle Villa at, is it the Peter Doherty Institute for Infection and Immunity? Is that where you're at right now, Michelle?
Yep.
Yep, there we go. Michelle used and the authors used a technique called metatranscriptomics, which basically allows researchers to identify activated genes, which helps to identify viruses even without having that base genome sequenced. The goal of their work was to quantify the diversity of viruses in Antarctic penguins, as well as their parasites. And this is important not only to understand viral ecology in extreme environments, but also to gain a baseline of viral diversity during a time when, say, the Antarctic Peninsula is warming faster than most places on the planet. So for this study, the authors collected cloacal samples from 3 pygoscelid penguins. So that's the Gentoo, chinstrap, and Adelie penguins. This is kind of a typical method used for sampling for viruses like avian influenza. The authors also collected ticks from under rocks and adjacent to penguin colonies and Paradise Bay. And to identify the existence of viruses, the authors targeted the viral RNA of the samples and used the combination of phylogenetic analysis and online databases to identify those novel viral species. The authors were able to identify a host of new viruses previously undescribed for Antarctic penguins. More specifically, they were able to identify 103 viruses in the samples, of which 13 were capable of infecting penguins. Now, the diversity of the viruses didn't seem to differ between sites. And I think it's figure 4 that kind of shows that there were 2 viruses that were shared between sites in Adelie penguins. At one site, a sample from Gen 2 penguins actually yielded a type of coronavirus, a Delta coronavirus. And Michelle would probably be able to speak more about what that actually is compared to the beta coronavirus that causes COVID-19. But as she states in the paper, these types of coronaviruses are common in birds all over the planet. Interestingly, the viral diversity identified in Antarctic penguins was similar to the viral diversity for birds in other parts of the world. And that sort of suggests that penguins experience similar pathogen pressures to avian fauna in more temperate or, say, tropical climates. From the ticks, the authors were able to identify 2 species of virus that could infect penguins. And if you go through the paper, figure 4 is really a nice one that kind of summarizes the viral diversity in each sample and between the penguin species, the sites, and the ticks. So there's quite a lot in the results of this paper on specific virus types. If you go through, if you have an interest in viruses, if you're working on viruses, you know, I highly recommend going through that results section. It actually is very well written, and it does describe each of these specific virus types in great detail. I won't go into it because obviously we're not here forever. We want to keep the discussion relatively high level and not break anyone with RNA and DNA jargon and all that sort of stuff that goes probably beyond my head. So Michelle, I have a bunch of questions for you. And first I want to thank you for this very cool work. I know you've been working on Antarctic penguins and viruses for a few years now. I've seen a couple of your publications appear across my desk over the last little while. And thanks for sending through that article, the PopSci article, because that really helped condense a lot of the stuff for me that, like I said, was kind of over my head. So it was nice to see that. That helped summarize things a lot. So I think in one point in that article, you mentioned that there was a virus identified on the peninsula that was previously sequenced from samples in the Ross Sea. And so my question around that is, well, is it possible that that's a virus that's mixed through the entire Adelie penguin population, or is there some other disease vector or viral vector transportation mechanism that would lead to that connection?
Great, thank you. First, I would like to say that you can do popular science renditions of any of my papers in the future that you'd like, because it was a very nice explanation. So pertaining to this question, the virus that you're talking about is a member of the Astroviridae. It's a virus family that is not really often spoken about. It causes gastro in kids. It's around, it causes a disease in chickens. So there are many species within this virus family. And a number of years ago, Ray Grimaldi and her team went and tried to sort of sequence viruses that may be causing something called feather loss in penguins. And so they didn't sequence the full genomes of viruses, they just sort of sequenced these little tiny itty bitty fragments. And so what we found is when we pulled out this virus, we have the full genome of it and we ran it through databases. What came back from the database was a match to this really short sequence from a paper. And so the penguins that we sampled were not diseased. They didn't have feather loss. They were fine. So I would speculate that this is not the virus that's causing that disease. It's just a virus of penguins. Now, the problem we have is that sampling is so incredibly sparse. We just don't really understand how transmission works of viruses in Antarctica at all, actually. We just don't know. So there's a few ideas. One could be that it's not actually a virus of Adelie penguins, it's just that we've found it only in Adelie penguins thus far. It could be a virus of all penguins. It could be a multi-host virus. We may find it in birds like skuas or sheathbills, birds that have a bit more mobility. And so I can't even speculate as to the answer to that, because I just think that at this point we have zero data upon which to draw any conclusions. Yeah, sorry.
It's fine. I think no data is still data, right? Or or zero is still data, I should say.
Yeah, so, there's a lot of scope for a lot of work to be done, I think.
Yeah. So the sites that you got your samples from looked like they were from near research stations primarily. So Paradise Bay.
Yeah.
And
so the person collecting or in charge of the sample collections is Daniel Gonzalez Acuna, who's from Universidad de Concepcion. And so the trips are often supported by the Chilean military bases.
Mm-hmm.
And so, for example, the samples from Copaitic Island, the team stays on O'Higgins base, which is a Chilean military base. So they're very closely associated with bases.
Yeah. So do you think is there any evidence that, say, some of the samples might be, I won't say contaminated, but affected by the fact that they're close to human research stations? Would the viral diversity be different in more isolated populations?
So that's a good question. Something we'd love to do is to sort of sample penguins that are in the middle of nowhere and not near anything versus those that are close to human habitation. Or actually, rather, the more interesting question would be penguins on the fringes of Antarctica that perhaps intermingle with other bird species that are coming and going a bit more versus those that are in more isolated areas. I would suspect that birds that are in really isolated locations, you probably find less viruses when they're there because there's nothing to bring a virus in to go through the colony. Whereas on the fringes, there's more movement. I don't think it's necessarily correlated to where there are people, though, because we're not finding mammalian viruses in these birds, we're finding bird viruses. And so the reason I know that, just to give you sort of an idea, is that if you take the sort of tree of vertebrates and the tree of the virus family, you tend to get a really similar shape. So you'll have a clade of related viruses that are found in birds, clade of related viruses that are found in mammals, and that's how we can pull out these viruses that we know are not bird viruses. They're from invertebrates or the microbiome, is they fall in really, really different clades. So it's definitely, at this point, doesn't look like there's any influence of human viruses in the birds. I think the bacteria story is different. So we found evidence of antimicrobial resistance with higher levels in penguins closer to bases with more people. Not surprising, but for viruses, we don't see that at this point.
Cool. So the other thing I had, the other interest sort of, in your paper, you say something about how only a certain number of these viruses can infect penguins. So you'll have to ignore the fact that this might sound like a stupid question, but how can you have identified 103 viruses, of only which 13 can infect penguins, from samples that you took from penguins? Is this like, are these viruses that only, say, live in the gut but don't actually impact or infect the blood? Or perhaps you could elaborate on that for us.
Sure. So this is something, when I started with this field, I grappled with. It took me a while to figure this out, but we're taking samples, cloacal samples, and so cloacal samples are also influenced by feces and whatever's happening in the gut. So the viruses we're finding there that are not viruses that infect penguins are viruses that infect invertebrates and plants and bacteria. So they're viruses of the microbiome, and they're viruses that are left over from whatever the penguins are eating. And so sometimes you get the full genomes of those, sometimes you just get sort of partial genomes, but from birds, I've realized that there are bucket loads of viruses that we're finding in these samples that are not actually viruses of birds, right? So as I explained before, if we look at the phylogenies, we see that they fall into virus clades of viruses that are from invertebrates or bacteria or things like that. It's something that's ticking away in the back of my brain. You know, I think after a few more studies, it's time to go back and sort of scrape up all those viruses to see what's going on.
Mm-hmm. Yeah, cool. All right, so then my last question before I hand it over to David to grill you will be about sort of the conservation perspective here and the tourism perspective. So when we go to Antarctica, we go on a cruise ship and we get down to a colony, we go off and we do a whole bio-decontamination. We scrub our boots down and scrub ourselves down before we get to the colony. And when we come off the colony, we always scrub our boots down again. Now, there's the idea behind that is that you can, you know, as people going down there, we're traveling between colonies, and we don't want to introduce new viruses or bugs that could impact the penguins. What's your sort of thinking on that? Do you think that because now we have a better idea of the viral diversity in these penguins, do you think they're probably more resilient to disease than we think?
So that's an excellent question. The idea that viruses are associated with disease is actually a very old idea, and it comes from the fact that viruses were only ever described when you had a person or a domestic animal that was diseased, right? So we have a sick person or a sick bird. We want to know what's causing it. We have found a virus. So that's been the thinking for a very, very long time. The research that I do is we go and we sample healthy birds, and we find bucket loads and bucket loads of viruses in healthy birds. I think you can go sample any wildlife and you're going to find bucket loads of viruses in them. They're going to be infected with multiple viruses at the same time, and they're not going to have disease. I think that viruses that cause disease are the outliers and not the norm. And I often write this into papers, because how can we find an avocet that's shedding 9 viruses at the same time and not be sick? Like, it boggles my mind a lot. And so I think that there is always a chance that you're introducing something that may cause disease, but the vast majority of the viruses out there do not cause disease, is my opinion. I think that being careful around colonies, you know, there's the chance of introducing people viruses into penguins is also really limited. Birds and people are quite different, but in a place like Antarctica, you probably are better safe than sorry. You know, what does it cost you to do a bit of extra cleaning to make sure that you're, you know, not accidentally doing something that you can't really measure? Yeah.
Yep. Lovely. Thanks for that. All right, David, do you have any questions for Michelle?
Oh, but first of all, yeah, that was really excellent, excellently told, Grant, and I really enjoyed listening to the explanations. That was very clear. A little bit along these lines, I also had the impression that in these sort of studies, wherever you sample around the world in wildlife, you do find many viruses. So that seems to be a constant. I was wondering, and that builds up a bit on what you said, whether it's actually the proportion of harmful or potentially harmful viruses versus all viruses in a species, which is interesting. And if that works, how do your results compare to what has been found with in penguins, for instance, in South America? If you would take a Magellanic penguin, of course, in areas where it's much more exposed, would you predict that the proportion of potentially harmful viruses would be higher?
All right, so first things first, watch this space. We have a paper coming out talking about how the proportion of disease-causing viruses in all vertebrate taxa are decreasing with increased virus
okay, yeah.
So hopefully we'll get that submitted really soon. It'll go on bioRxiv. If you look at penguins, disease is pretty rare. It happens sometimes. We have done a study recently on Magellanic penguins. I'm a co-author with some folks from Sweden, and we have found multiple picornaviruses. These are viruses known to cause gastroenteritis in poultry that have caused disease in these penguins. And we have a number of individuals that are shedding 4 different picornaviruses at the same time to crazy high levels. So the idea really is that disease is the exception. It is not the rule. And I think no matter where you sample birds, you know, viruses causing disease are very rare, and I suspect they're due to recent cross-species transmission events.
Okay, so I was spot on with choosing the Magellanic penguin as an example.
I was really pleased. I was like, I know the answer to this. I'm working on it right now.
Yeah, Yeah, no, it just struck me as the art of penguin going around in really filthy places.
Yeah, you know, we've done some sampling of ducks in sewage treatment plants and in more pristine areas, and they have the same number of sort of viruses, and we don't see more disease in ducks hanging out in sewage plants as compared to others. So,
and you mentioned this briefly, and that will be my last question. You mentioned this briefly before, but is there anything special with birds compared to mammals? With respect to, you know, how many viruses they may carry without being sick? No specific resistance in birds?
so I don't think there's any special resistance in birds. I think that most of our data on viruses causing illness come from poultry and from cows and pigs and people. If we remove any animals that we mass produce, we remove those from the equation, you also see that many mammals do not have overt disease when carrying viruses. I mean, let's just think about bats, for example. Bats carry some of the nastiest viruses, and by nasty to people, and they show no signs of disease. There is a lot of argument that bats are special, but I would perhaps argue that people just started thinking about this in bats before other animals, because of these nasty things like Ebola. I think you could sample really any mammal and sort of find that most viruses don't cause disease, but if a virus from a buffalo goes into cattle, then you're probably going to get disease.
Oh, fascinating.
Very cool. All right. Do we have any questions from the audience? Nobody's raising their hand at the moment, so I'll ask you. Oh, actually, Eric's got a question. Hey, Eric, hang on a second. I'll unmute you. There you go. Go for it, Eric.
Thank you. Michelle, great paper. I really enjoyed it. It's kind of fun when you mentioned Ray Grimaldi, because she did her honours degree with me more than 10 years ago, looking at this question of disease in Antarctic seabirds. One of the things that we raised in her honours thesis and publication was the fact that we now have professional tourists that are quite happy and quite capable of getting from the Arctic to the Antarctic in less than 24 hours, and potentially with less than the most stringent of biosecurity issues. So even just parking the issue of introducing disease, there is also that sort of parallel issue of altering the biodiversity in terms of just simply introducing species that can become established in analogous habitats from polar regions. So I'm just curious if you know, as I said, parking the idea of disease, but just simply the fact that, is there any evidence that some of the viruses, whatever that you're finding, may have actually originated from other parts of the world that have been introduced to the Antarctic?
I think we are utterly unable to answer that question. Before our study, there were about 15 described viruses in Antarctica. We essentially have doubled that number with one paper, a handful of samples. I think that we have utterly no clue of what the true diversity is in Antarctica, and we cannot even begin to speculate what is introduced until we have baseline data. I think that yes, their viruses do live on surfaces. It can happen. Things like the coronaviruses are pretty hardy. But I think it's difficult for people to be introducing viruses from the Arctic to the Antarctic, even through airplanes. It can happen. Don't rule out anything, but I think that it remains sort of mostly unlikely. Maybe they've got some contaminated calipers that they use twice only on penguins and have never cleaned it between, which would just be bad form. But yeah, for me, we don't have baseline from the data from the Arctic. We don't have baseline data from the Antarctic. How could we know? And it's concerning because I read a lot of papers about, anthropogenically mediated virus introduction or climate change mediated viral change. But how can we say anything? We have really no clue. And I think if we redid this study with the sample sets from 3 other years, we'd probably double the number of virus species again. You know, we're not.
And very naive question: how far back can you go? So can you only trace viruses occurring right now, or is there any way to find archives?
So the problem with sampling in Antarctica, this one hurts my brain when I think about it, is that there are problems with freeze thaw, and so samples that are old that I've tried to test from archives were not necessarily treated really well. So some of these viruses are quite delicate. You need to take the sample and you need to put the sample in a -80 freezer, and you cannot let it thaw and freeze and thaw and freeze. If you do that 4 or 5 times, there's garbage left in your sample. So, you know, Aaron Hurt, who I worked a bit with on this stuff, asked me to screen all these archive samples. And if we actually look at what the RNA purity and or quality is it actually quite garbage. So if there is a sort of an archive set of samples that exist that have been really well taken care of, haven't undergone freeze-thaw samples, yes, we can go back to archive samples for sure. But I find even with recent sample collection, some years the quality is just not very good, and it's probably because there wasn't a good freezer at a place, or there was no power for a while, or something like that.
And samples not in freezers, but actually in the field. Is it conceivable?
So with the idea of MinION and sort of on-site sequencing, I think there is potential. We find that the MinION is not very good for the kind of work that we're doing, because so essentially we take a swab from a penguin and we sequence everything. 99% of what we sequence is penguin. So we throw that away and we look for what's left. And so these sort of sequencing technologies that you just plug into your laptop and go, they don't work well for that. They work well for sequencing what's common in the sample. But that doesn't mean that in 5 years they want to solve that. So we can probably sort of look to do more lab work in the field in the future, which would unlock a lot of the sample condition problems that we have from Antarctica even today. Yeah.
Cool.
If I can.
Yeah, go for it, Eric. Sorry, I didn't see your hand there.
That's okay. Michelle, and just to continue on from David's question, there's 2 wonderful natural biological archives in the Antarctic. One comes from snow petrels when they vomit their oils, and so you have Mumio that is literally thousands of years old, nicely layered, beautifully able to be dated. And you also have the relict pygoscelid penguin colonies, where people like Steve Emsley have identified and dated material going back 10,000 years. So I suspect both of those might be fun places to start playing with.
Yeah, for sure. And I think that, you know, therefore viruses that sort of have hard shells like the coronaviruses, you'll probably be able to manage. But for sort of the soft, delicate viruses like influenza and coronaviruses, you're probably going to have a bit of a hard time, because they don't do well with not being inside cells and inside bodies. I think there's a ton of scope for work like this in Antarctica. You know, we've just scratched the surface, and it is due to cost and logistics mostly.
Of course.
Yeah.
I have one last question, I think, if no one else does, which is actually related to a paper we reviewed last week when we had David Ainley on. There's a Santora paper that just recently suggests that these colonies actually occur in clusters, and that they're closely related clusters that are based around, say, a source population. So what do you reckon that it's likely that the viral diversity might be similar between colony clusters? Could that be a way of identifying colony clusters?
Yeah, super interesting question. One of the big flaws of our study was that we couldn't really control, or I couldn't really control when samples were collected. So it's a bit hard to compare between species and things like that. But I reckon if you had colony clusters and had something interesting that went in, it would run right through the population. So if we could control for sampling better, it'd be great to employ a lot of the community ecology analysis, which we did a little bit in this paper, but quite ineffectively because the data structure wasn't great. But yeah, you could probably identify colony clusters through viral sharing for sure. But having said that, Adelie penguins are pretty weird, because in this study and in sort of the study that came out last year in Journal of Virology, we found that Adelie penguins on Kapaitik Island and even on the South Shetland Islands are shedding the same viruses, the same things. And we're, maybe they're just really good hosts for these viruses and they usually always have them, but it seems strange to me that these really distant colonies were shedding the same virus. And so there must be sort of, I don't know enough about Adelis to understand how much sort of transient movement there is between colonies of juveniles or things like that. So that might mess a bit with the idea, but you could. I think it's a great idea.
All right, I think with that, we'll probably end it off and let everyone go about their evenings, days, nights, afternoons, whatever it is they've got planned. And Michelle, thank you very much for joining us. That was a really cool read.
Yeah, thanks, Michelle. That was really interesting.
Yeah.
Yeah. No, thanks for having me.
And very clear.
Yeah.
Great.
Yeah. Very well written paper. So thank you very much. All right, everybody. I think with that, we'll say goodbye. I hope you all take care. We'll get back to you as to timing for next week. Normally we would be at the 1600 slot, but we're just trying to sort out logistics around whether or not we'll be able to get David on from next week, where he's going to be off in the field playing with birds, which all of us wish we were doing. So, but we'll be in touch. So keep your eyes to the email list. If you're not on the email list and you want to be on the email list, please just drop me a message. And soon I'll be circulating a quick survey, a Google form survey, to get feedback from participants on Seabird Sessions, and if there's any ways we can improve or streamline the process, make it more engaging for everybody. So with that, I hope you all have a fantastic evening, day, have a fantastic week, have a good weekend, and we'll see you next time.